Chapter 5, the mitotic cell cycle. Under that, we'll start off with the very basics, where we discuss about chromosomal structure. I'll give you a very small introductory point, and this is a very vague way of describing what a chromosome is. So I'll say that a chromosome is made up of long strands of condensed DNA. Condensed in the sense packed. Condensed DNA molecules. And they are specifically wrapped around something known as a histone protein. Now, you might have an idea of what a histone protein is. You might not have an idea of what a histone protein is. That is perfectly fine. You will get that context under this topic, histone proteins. Right. A picture is worth 1,000 words. Now, I'll start off by giving you a rough sketch of what a chromosome looks like. Now, bear in mind, I'll give you a small disclaimer before we begin. Chromosomes are very structural, very complex molecules. Okay, this is going to be one of the most vague ways in which you can draw a chromosome, but I have to tell you to appreciate the biology behind this. Chromosomes are much more complex than this diagram makes them seem. Right. Basically, that looks like an X. I'll actually draw both of them in equal manner. Whoops. Then the below strands. Nope, that came off too much. My God. Why do I keep repeating the same mistake? Okay, that's better. Right. So let's start off by labeling the pieces. I'll scroll down. First of all, this centerpiece module is referred to as a centromere. Is referred to as a centromere. This is our entire chromosome. And each of these individual strands is referred to as being a chromatid. Chromatid. So if you were to picture it like this, a chromosome actually contains two chromatids. Contains two chromatids. This is the first chromatid. This is the second chromatid. Now I left some space over here. Any idea why? Whenever we are referring to both chromatids in a chromosome, we refer to them as sister chromatids. So a single chromosome will contain two sister chromatids. Sister. For some reason, on a biological perspective, they like to use female names. So, mother cell, daughter cell, sister chromatids. You will not find brother chromatids. You will not find father son cells. It's always going to be a female perspective. Then I'll talk about these little ends. Now, I'm going to cap these ends off like this, and I'll give it a little bit of a shaded outlook like this. Okay, this is going to distinguish the ends of the chromosome. Right? The ends of each chromatid, therefore the ends of the chromosome. These ends are referred to as telomeres. Now, telomeres have a much more important function than you might think. But for now, all you have to know is these things are known as telomeres. The end parts are known as telomeres. Now, what about this histone protein thing? Now, I clearly told you that DNA is going to be wrapped around this histone protein. Now, you can't really picture that out of this. This is a condensed chromosome, so I'm just going to take this part and uncondense the DNA. We know DNA looks something like this, like so. And we know around the specific proteins only DNA is wrapped. So throughout a regular interval between these DNA strands, we're going to have these little proteins. Around those proteins only will you find DNA being wrapped, and these are your specific histone proteins. Your histone proteins. Right? Now, when DNA is actually wrapped around these histone proteins, we call it chromatin. Now, I'm going to introduce you to a small distinction. Now, this is actually something known as chromatin. All right? Now, this distinction is not really required at IGCSE levels or even sometimes AS levels, but this is going to be very important for you going further to properly understand the nomenclature. DNA itself is actually the condensed form of chromatin. There's more to that, but very vaguely, very roughly, if I mention, when you combine chromatin with histone proteins, you get DNA into the packaged form. Those are the basic parts of the chromosome you should know at this level.